All right, so for this video, we're going to look at transports across the membrane. And the first one we'll look at is called passive transport. And what we need to remember with passive transport, it's the movement of molecules from a high concentration to a low concentration, and we do not eat energy. We don't use ATP, we don't use any form of energy, we just go down a concentration gradient, high to low. So with passive transport, we have a couple types of diffusions. The first type is simple diffusion. Simple diffusion is used for gas movement. So oxygen, carbon dioxide, small molecules like that can move across our membrane easily. No protein channels needed, no energy is needed. It's just a free movement across until it gets to equilibrium on both sides of that membrane. We then look at osmosis. Osmosis is just a free movement of water across the membrane. That again doesn't need much, just needs a concentration gradient until it equalizes itself. Okay, some of our cells do have what are called aquaporins, which are little specialized channels for water to go in and out. But for our sake, we're going to look at water and just say that it moves across that membrane freely until it meets equilibrium. We have facilitated diffusion, which is over here. Facilitated diffusion, you look and you see these little gray looking spheres. Those are protein channels. That means these molecules are larger and they need to be regulated using these protein channels. So glucose or larger molecules that have to move across from high to low concentration will go through a very specific protein channel. These are two different types. Doesn't really matter to us. We're just talking about protein channels here and the movement of molecules from high to low concentration because they are usually larger in size. Okay. Our last type of transport on this slide we call filtration. Filtration, you're moving from a high to low pressure. So inside of our arteries, we have a high pressure because that's what blood coming out of your heart is in an artery. So you have high pressure here, that's going to move to low pressure and filter things out. As you move along, you're going to get no difference from inside and outside of these blood vessels. And then as you move further down, let's say to our veins, you're going to have a lower pressure inside. So you're going to have stuff from outside with a higher pressure moving inside of these blood vessels. So you have high pressure inside, so things are forced out, and you have a low pressure inside, so things are forced back in. So you can think of with arteries, you're transporting glucose, oxygen, nutrients, minerals, anything like that. That's going to be forced out of the blood and into the cells. And then your cells use the energy, they produce waste. So waste is going to be coming back in. You're going to have carbon dioxide, things along those lines coming back into your blood to then be released in the from the lungs, breathing out carbon dioxide, or getting filtered out in your kidneys as well. Okay, so that's your passive transports or your simple transports here. Then we move to active transport. Notice this is not a diffusion. So everything in the previous slide didn't need energy, moved down a concentration gradient. Here with active transport, we're moving against the concentration gradient, so we're going to increase the usage of energy or ATP. So if we look here, we have a low concentration, we have a high concentration. What we want to do is get everything on one side of the membrane to really mount up a larger charge on one side. And when we get to the sodium-potassium pump in the nervous system, it will make more sense as to how or why this is necessary. Okay, so for active transport, for right now, we're moving against the concentration gradient, so we're going from low to high. And we're going to need energy to do that because it's not going to move through diffusion, because diffusion would want to go the opposite direction. So active transport, low to high using ATP. Your last type of transport for us right now is exocytosis and endocytosis. Exo meaning exit. So with vesicular transport of exocytosis, you're going to take one of these vesicles. They have we're going to talk about neurotransmitters being released from one neuron to another neuron. And this is one way in which they do that. They take this vesicle that has some type of molecular compound in it. It attaches to the edge of the membrane, and then it spews out those chemicals, okay, or hormones, or whatever you want to call them, neurotransmitters. So that's exocytosis. And then we have what's called endocytosis, endo meaning bringing in. And endocytosis, you're going to encapsulate it around the membrane and you're going to bring it in as a vesicle. We look at this the most in our white blood cells, which we call phagocytes, or one of our white blood cells is phagocytes, and it's through the process of phagocytosis. And that's the enveloping of large, let's say bacteria or viruses or whatever it may be, to then try to destroy them. So if we look at this picture down here, here's the bacteria. We're going to engulf around the outside of it. We're going to bring it in. Our lysosomes and other cell-killing chemicals are going to destroy it. And then we would get rid of that debris through exocytosis, actually. And then that would go into our bloodstream, and our filters would, our kidneys would filter it out and get rid of it out of our system. Okay. So endocytosis is a general term for enveloping molecules inside or items into a cell. Phagocytosis is specific. We're going to see that more than anything, which means we're enveloping large, usually bacteria or viruses inside of the cell. Okay. So I hope that helped. And if you have any questions, we can obviously go over it or talk about it in class tomorrow.